This is episode 306 of the Stem Cell Podcast, Vertebrate Development with Dr. Carole LaBonne. Hello, everybody. This is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. This podcast is brought to you by Stem Cell Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, as I said, we have Dr. Carole LaBonne, who's from Northwestern University, and she's on the podcast to talk about research on neural crest differentiation, as well as other things. We've also got our usual roundup of recent highlights in stem cell news that's coming right up.
But first, we'd like to remind our listeners about ESC and IPSC News, one of Stem Cell's free bi-weekly scientific newsletters. ESC and IPSC News summarizes all the latest research, news, jobs, and events in ESC and IPSC research, and delivers it right to your inbox every other Wednesday. Save time and keep current with ESC and IPSC News. Subscribe for free at www.eslnews.com.
Just before we start the roundup today, a quick bit of sad news for the stem cell field and the world. And apropos here around Nobel Awards, we lost John Gurdon, who was a bright light in the field, had to wait decades before he really got his flowers. But the OG developmental biologists and all of us really, those who care about the work have always known that name. It's always rung out. My graduate school mentor was trained directly by Doug Melton, who was trained by John Gurdon. My current chief almost did a sabbatical with John Gurdon. He's touched so many scientists' lives and really influenced the field. We interviewed him on the show during an ISSCR meeting a few years back, which was quite honestly and sincerely the highlight of my career as a scientist, not just as the host of this podcast. Rest in peace, Dr. Gurdon. You really changed the world and invented the thing we talk about. So, all credit to you. And up there, you're doing experiments now with a different substance.
Yeah, I mean, it's such a tremendous loss for the field. He is, and he's been inspirational to all of us, right? I mean, if you work with IPSCs, his work is the reason why you work with IPSCs, because his work ultimately led to the discovery of embryonic stem cells, induced pluripotent stem cells. He's truly foundational to the stem cell field, and there's basically 2 or 3 degrees of separation for all of us in the lineage of Dr. Gurdon. Samantha Morris, who was on the show not too long ago, was very appreciative of his mentorship. Um, he will definitely be missed, and I'm sure his loss is something that's going to be highlighted at ISSCR in the near future. So yeah, tremendous loss for the field.
Yeah, and Sam Morse actually had a great anecdote, I think, that really spoke to me about his commitment and persistence, where she said, she talked to him at age of 73, he was rushing right from the bench. And I remember when we talked to him, he was still doing experiments. So I hope I can continue to be as not just productive, but as passionate about my work as he has been for, pushing 9 decades. Anyway, to the roundup. This episode, we have a lot of stories surrounding, maybe you could argue, the foundational models that led to all this work we're doing in human, in large part done by Gurdon, and in his case, in the frog and beyond. But we're talking about the whole stem cell zoo today. I'm starting with a story that's about haploid embryonic stem cells, but is actually really related to livestock and ruminants. And that's because, why this is of such interest. is cattle, sheep, which are the ruminants we're talking about today. They're a huge part of our food supply, right? Meat and milk. And genetic engineering of livestock. I batted this around a bit in a collaboration that almost got off the ground, didn't get the funding with the veterinary school here at Cornell, and I learned a lot. That genetic engineering in livestock is really one of the keys to improving ruminant agriculture. But it's tough, right? Because genetic engineering, there's two alleles, right? And that's where the haploid stem cells come in. So haploid androgenetic embryonic stem cells, they contain only one set of chromosomes. And the way you make them is you enucleate an egg and you just put in a single sperm, and then that develops into blastocysts. You derive stem cells from that, and it's a haploid stem cell line. And you can inject these haploid stem cells by ICSI into an egg in rodents, and they will like act as a surrogate of sperm and fertilize the sperm. So they're kind of like a potential replacement for sperm clinically, potentially, although we could talk a lot about how I don't love that idea, in humans at least. But also really useful for loss of function screening assays. And that's because there's only one copy of each gene, right? So the phenotypes really jump out at you. Um, Now, haploid stem cells have been generated in rodent, and a lot of the work has been done in that model. But in larger animals, it has not really been recapitulated, particularly in livestock. These haploid embryonic stem cells have not been generated. And this story was a tour de force from a technical standpoint. And you really have to look into the story to realize how much work and innovation was involved in this. And of all places, came out of, in large part, there's three lead authors on this: Lei Yang, Xiaorong Gao, and Guangpeng Li. Two of those are from Inner Mongolia University. You didn't think there's anything coming out of Inner Mongolia, but let me tell you, agriculture. So Inner Mongolia contains like a third of all of the grassland of China and a quarter of all the pasture land, and is famed for their livestock, right? So clearly, their scientists are motivated by that endeavor. And the group here, what they were going after was the first to generate these haploid embryonic stem cells. They did it in cow and sheep. And I mean, it's pretty traditional. And you want to look at the technical elements, look at the story yourselves. This is a Nature Biotech paper article. Um, but the what was so impressive to me, so just going through the details here. They made the haploid ES cells. They showed that they could generate offspring using this intracytoplasmic haploid ES cell injection, which they called ICHI as an acronym. Um, and just, talking about the characteristics of these haploid ES cells, they are just like you would expect. They represent this formative state of pluripotency, or that's the correlate in diploid ES cells. They differentiate into three germ layers, in vitro and teratomas. But the key here was how they got them to generate actual conceptus in live-born animals, and that's because it's tough, right? It's not just a sperm surrogate. It's a haploid cell, and because it's haploid in all of its ontogeny, it lacks the correct methylation pattern, right? And this has been well described in the rodent system. That, like the rodent haploid ES cells, the embryos that they made from this Ichi process, intracytoplasmic injection, they couldn't develop to the blastocyst stage as is. And it's while it's previously shown that there's like aberrant methylation and also of DNA, also of histones, and that's been a barrier to progression of those embryos. Here, they used the classical methods. They did this whole descriptive analysis and bisulfite sequencing, all that, to show that it wasn't aberrant methylation of DNA. And then they went after the histones by multiple methods, ultimately settled on protamine, which was able to allow these embryos to progress. and get full term development of these embryos. And then uh, m- they combined this protamine facilitated intracytoplasmic injection and prime editing uh, to generate cows and sheep that were gene modified. So, like, soup to nuts, overcoming these real technical obstacles that have clearly been a challenge for many groups who I'm sure have been after this. This is a pretty heady field, and then going all the way here by showing that the real, you know, ultimate case use of generating these gene modified animals here in a manner that's like really straightforward, doesn't involve a lot of breeding. It's a huge shortcut. Um, totally feasible. So here we are, straight out of Inner Mongolia. They're doing it over there, partner, and setting the stage for a revolution in the way we breed or generate livestock, at least.
Straight out of Mongolia. I love that. I bet it yeah, you're right. I mean, Mongolia, Inner Mongolia has been at the forefront of livestock for literally thousands of years. I mean, it's the steppes, right? It's some of the most fertile land, I suppose, or, usable land for livestock in the entire world. Feeds the country of China in a lot of ways, right? Um, it's really an exciting technology and advance and very worthy of being in nature biotech, that's for sure. And for me, it's about the implication. It's, the ability to produce these genetically modified livestock using technology such as prime editing, right? This next generation version of genome editing, which is even better than CRISPR. You could just think of the implications here. They're tremendous, making animals with improved disease resistance, improved productivity traits, I don't know, different physiological features and looks and reducing environmental footprints. I don't know. There's so many other applications that you can think of for this work. I mean, this is really groundbreaking stuff coming from Inner Mongolia.
Yeah, and the Black Mirror element of this, not the best analogy, but you just described all you know, the overt applications, but it gets me thinking about even the fact that this is so, the timeline, right, and the amount of effort and the scale, volume, multiplicity of edits that you might do says to me that, like, hey, are we now moving into this next generation of agriculture and genetic engineering of our food supply? Uh, you know, garden variety fortification of milk, or next level type stuff, where exotic type stuff that who knows, just let your imagination run wild. We're in a new era, Arun, and it's crazy. Inner Mongolia leading the way.
Leading the charge, leading the way. Absolutely. Very, very cool stuff. And like I said, one of my favorite parts about this story is, the fact that you're using prime editing technology that was described by David Liu and colleagues within the last 5 years. We actually covered the initial prime editing story on the Roundup. I remember that. And here we go. It's being applied in Inner Mongolia, of all places. Moving on to another story in this theme of animals and stem cells for today's roundup. Um, I'm going to be talking about the derivation of embryonic stem cells across avian species. This is also a Nature Biotech paper, and that's pretty much the paper. It's the creation of ESCs, bona fide ESCs, from a lot of different avian species, including the chicken, from the turkey, and just a bunch of different species here. And again, a lot of implications here for livestock, but also for biodiversity and birds. And I'm for one, I'm kind of surprised this hasn't been done yet. I think iPSCs have been derived from different bird species, but ESCs have been notoriously tough. I mean, of course, germline competent ESCs have been derived from a number of different species, mice, rats, mammalian species, whatever, but not so much from avian species. So here they reported the development of good culture conditions, appropriate culture conditions that's enabling the derivation of embryonic stem cells from chickens and seven other different birds. So the chicken ESCs express core pluripotency markers, as you might expect, to validate the production of the ESCs. They can do their standard differentiation into all the embryonic germ layers, extra embryonic lineages too. And also notably, the chicken ESCs were able to contribute to high rates of chimerism. So another way to validate the integrity and the stability of these ESCs, the potency, right? So when they were injected into the chicken embryos, they gave rise to germ cells of both the in vitro and in ovo as well, which is able to confirm their germline competence. They're able to show that ES cell self-renewal pathways are actually conserved across avian species. So you got that evolutionary development angle there. Um, and This allows ESCs from different avian species, different bird species, to be actually established using optimized chicken ESC culture conditions. So you can use the same chicken culture for culturing the ESCs from these different bird species. And that makes things really cheap and easy. And actually was diving into a little bit of the backstory. One of the key factors that was actually unlocking this ability to have this custom culture condition across species was using egg yolk. So they had just added chicken egg yolk into the culture media and showed it contained some critical factors that was able to maintain the pluripotency network. So that's pretty genius if I have to do say if I say so myself. So um, yeah, and a lot of implications here. Like I said, future applications for genetic engineering, avian biodiversity, conservation, potentially livestock applications. Although I was talking to my mentor, Paul Burridge at Northwestern, who's been on the show a while back, who actually has his own company focused on pluripotent stem cell derived meat products, clean meat products. He says that the livestock and meat production implications here may not be as great because you know bird in vitro cultures for consumption, that's already pretty cheap and easy. So I think his startup that he has is actually not focusing on ips derived chicken nuggets, for example. um, but yeah, regardless, the implications are definitely there for looking at biodiversity. And again, the broad range of species they're able to look at: quail, goose, duck, turkey, pheasant, peafowl, ostrich. And the fact that you're able to have a common culture condition, common culture media for all of them is pretty exciting. So this is a pretty heavy lift, but a pretty important one from the lab of Li Shilong Ying and Carlos Luis also on this paper. So yeah, really exciting stuff. Again, following in the theme of the first paper there.
Yeah, this really took me back. I loved this story. It was inspiring to me because I mean implications and all that great. You said it. But the the way they went about it, right? It was like, old school, the alchemy type, witch's brew. And you talked about the yolk. I love the way they wrote it out. A lot of times you read these stories and you know that it didn't happen that way. You know that like they're telling the story back and forward. But here they're, yeah, we noticed that the ones that had a little bit of yolk did better. So we did it in yolk and it worked. And then that's when the lift comes in. They do the fractionation and there's all the other factors and they find that it's ovotransferrin. Like that's the reason why no one's ever done it before because it's tough. A lot of people have tried, but just like that previous story with the haploid ES cells, like, yeah, you think it didn't occur to anybody that that would be a lot more of a benefit in ruminants than in rodents. I mean, we're not trying to eat rodents. And similarly, I'm sure a lot of people have gone after this, but it really took the deep dive and the nitty gritty, right? The hard work, the science, and so I read a story like this, and it brings me back to method, right? Just follow the data, take that lucky observation, that one case that came through, and just drill down and figure out and deconstruct, get that mechanism. This is a great illustration. They should teach this paper in class.
They really should, and they should teach it in our master's class here at Cedars because it's work that's happening just down the road from us at USC. I didn't realize that the last author here is actually at the Broad Center for Research at the University of Southern California. Um, Really ingenious, and like you said, that brute force effort and that kind of teasing apart of individual mechanisms, that's how iPSCs first came into play and first were discovered, right? It was just Yamanaka and, just teasing apart the individual, transcription factors that were critical for that, reprogramming process to occur. So, glad to see the scientific method is alive and well, even in this modern age of AI. Um, really cool to see this work.
Yes. And hey, get me all them foul ESLs. I got another story here, although we're taking a little drift away from the stem cell zoo here. But kind of related to my previous story about the haploid cells, although this is really talking about sperm, the cell of origin for those haploid ES cells, although maybe a little existential twist here, more related to humans and genetic drift. I will elaborate, right? So this is kind of scary facts here that I didn't know. The genome of the average newborn has roughly 70 de novo point mutations. All right, think about that. Just roll the dice 70 times, one in a billion or something. Could be bad. I'll take those odds. But roughly 80% of those, and here's the key to this story, arise in the paternal germline, right? And this can be a cause of sporadic monogenic disease if the odds work against you in those rare cases. And this idea of the disproportionate allocation of these de novo mutations to the paternal genome has been the impetus behind a lot of these large cohort parent-child trio studies, right, where you take unaffected parents and their children who could be affected by a range of conditions. A lot of times it's neurodevelopmental disorders, autism spectrum disorder, but could be anything. There's been other studies looked at congenital heart disease, etc. But within these studies of de novo variation, which, as I said, most prominently are in these sporadic neurodevelopmental disorders and autism spectrum disorders, there have been hundreds of disease genes that have been identified. Perhaps um, most notably was this Apert syndrome, the gene underlying Apert syndrome, and pretty famously linked to this paternal effect because it involves this spermatogonial clonal amplification, which I'm gonna come around to, right? So there's a small set of these hundreds of genes that have been identified by these trio parent-child trio studies, but a subset of those defy logic, right? There's like these algorithms and models of how mutations should emerge by the numbers. And there's just few of these genes where it's like, yeah, that's disproportionate. And one of the explanations for this is that there's other forces. There's propensity beyond just random mutagenesis that increases the observed incidence of mutations in these genes, right? So so-called hotspots, right? And a lot of the genes within this subset that have been observed are involved in carcinogenesis or clonal expansion in somatic tissues, which may be benign or malignant. Um, and that suggests, coupled with what we know about Apert syndrome, which I haven't elaborated on, but it's this idea. It suggests that there may be a positive selection of 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 those mutations that contributes to a clonal expansion in the spermatogonia. And these clonal expansions, right, uh, if they are essential genes, can lead to monogenic disease. But these clonal expansions can be relatively benign and masked, right? Um, but unlike somatic tissues, where you can have clonal expansions, like take CHIP, for example, clonal hematopoiesis of intermediate potential or indeterminate potential, unlike somatic tissues, when you get a increase in these clonal expansion of the spermatogonia, you have an increased likelihood of transmitting those driver variants to the next generation, right? So you're going to get elevated incidence of these observed mutation rates in the offspring. And we're talking about just like humans across the whole landscape as fathers age. And as I said, early drivers of this clonal expansion and spermatogonia were identified because they contribute to monogenic disease, like Apert syndrome. But there's other genes that drive these clonal expansions, so these loss of function or gain of function mutations that drive clonal expansions that don't directly cause disease, right? But they will manifest, right? Those mutations, even though they don't cause disease, are going to be there. Um, so, what this story is about, which is a nature story, this is very deep in silico theoretical, but it has a very real component to it based on the data. It's from Shamil Sunyaev at the Brigham and Williams Hospital, uh, associated with Harvard Medical School. Um, they did an analysis of almost 55,000 of these trios that were in this rare condition uh, group. Then they had roughly 6,000 control trios, so healthy child, or unaffected child. And then they compare this to this database of roughly 800,000 in just the general population, healthy individuals, right? So massive deployment of the data here. And they found that there were genes that were manifesting these de novo mutation rates that weren't consistent with the models. Again, these outliers, and they identified there were 23 genes that were hypermutable, leading to loss of function. There were 17 genes that were hypermutable, like missense mutations that presumably led to gain of function. They found that the average mutation. So when you look at a population level, the presence of these mutations, if you go by the numbers versus what's observed, the loss of function mutations were observed at 17-fold what you would expect in the general population. And the gain of function mutations were observed at 500-fold what you would expect according to the models, right? So uh, this is real, is the bottom line. And what's worse is they found that this positive selection in the germline uh, increase the prevalence of genetic disorders in spite of the fact. So, what you would expect is that there would be a negative selection in human populations, right? Because these are like disorders, right? They should be selected against. What they found is that in spite of this expectation and trend in a normal, steady state, right, all things being equal, in spite of that, they found that these things were increased. So the observed incidence was increased, masking what you would expect effectively. So the prevalence of these driver mutations are widely extant in the human population. They are arguably being exacerbated in their incidence by increased paternal age. One bit of, I don't even call it good news, but they looked at that of 19 of those loss of function driver candidates, only 9 of them showed a clear link in terms of causality to disease. So, like, maybe some of these, clonal expansion driving mutations are benign, but, if half of them are causing disease, I do not like those odds. I was talking with Arun before the show about, I don't know, part of me is like, so does this mean that like we are just progressively degrading as a species driven by this clonal expansion within the spermatogonial compartment? And if so, like, we didn't talk about this, but I'm thinking about increasing plausibility of that movie, Idiocracy. Do you ever see Idiocracy? I feel like we're just getting more screwed up
Hmm.
as we progress as a society and species. We need to outbreed with the quickness.
Yes, sir. Um, call to arms right there. Yeah, I think a lot of interesting implications here, especially as folks get older and they have kids older and guys have kids older and in general, the overall fecundity of the human species, which seems to be declining and more rapidly in some places in the world than others. But hey, I'm going to be the optimist here in saying we have the technology to perhaps alleviate some of these things and through IVF, through I don't want to say genome editing because obviously that has other major implications for the species as a whole. But we have the technology to perhaps predict, identify, alleviate some of these concerns through things like IVF, perhaps. But yeah, I'm done having kids, I suppose. I'm glad I had my kids somewhat early. Um, yeah, good days, bad days, whatever. Yeah, a little bit of everything here. This story has a lot to think about. Let me put it that way.
Yes, that's for sure. It's dark. I mean, it casts a pall on my whole day. I need to forget about it as soon as possible. But Arun, I mean, I understand. I work with IVF, but I can tell you that it's not as fun as the alternative, my friend. So my advice to your young bucks and my own is don't wait. Don't wait. Find the one and, carry on the species for God's sakes. It's not just about you.
I'll finish the rhyme for you. Don't wait, mate. How about that? You
Brilliant.
guys can edit this out if you
Is that
need
a
to.
haiku?
I don't think so. Man, moving on to the last story of the roundup. This has nothing to do with human reproduction. This is about dogs, which are probably the higher order vertebrates at this point in time. Everybody loves dog. Not everybody loves humans anymore, but we all love dogs, don't we? So this is a stem cell reports paper, and perhaps they're going to cover it on their podcast. Last author here is Shingo Hatoya from Japan, Osaka, Japan. Signaling pathway based culture condition improves the differentiation potential of canine iPSCs. Again, going along the lines of our stem cell zoo that we're talking about here today: birds, bovine, human, and of course canines here. Canine iPSCs are useful. They are useful for modeling disease in dogs. Dogs themselves are valuable translational disease models, of course, with the recent shift towards non-animal model systems. Perhaps, of course, they're being phased out. But regardless, canine iPSCs can be useful for studying development um, in dogs, and there's still really a need for the appropriate culture conditions for dog iPSCs. I don't know anything about this field. Perhaps I should talk to Paul about this, but apparently it is tough to grow dog iPSCs. You have to have custom medium formulations. Differentiation protocols are not great. And so they established this group in Japan. They established Nanog Reporter canine iPSC lines and found that FGF, surprise, surprise, and TGF beta and Wnt, all of our favorite things here, are critical for the robust maintenance of these canine iPSCs. And so um, they had their custom culture conditions. They were able to manipulate these signaling pathways and showed the stable culture of these dog iPSCs, regardless of the cell line. So that's important. You don't want a media condition that's only working in a single cell line. And unfortunately, I've been in situations like that. Um, so, regardless of the cell line or the basal medium, you have these critical signaling pathways that can be manipulated to stabilize the culture of the dog iPSCs. And they showed in this optimized medium that the dog iPSCs across multiple lines had a homogenized global gene expression pattern. So, they're pretty standardized. That's great. Um, and also, the cells grown in this medium were able to differentiate into different lineages, including my favorite lineage, into dog heart cells, dog cardiomyocytes. Exciting. And again, homogeneous contraction, good sarcomere alignment. And so this is, again, shout out to Paul Burridge. I hope you're listening to the show. This roundup's all about you. But Paul would be happy about this because this is development of a robust culture condition, a robust culture media that seems to be pretty effective in growing dog iPSCs across a bunch of different cell culture lines, IPS lines, and you can use them for a bunch of different applications. I'm not sure about the applications for the dog iPSCs for, um, you know, dog reproduction and chimera studies and all this kind of stuff, but definitely for modeling, I think this is definitely very useful. So I like dogs and I like iPSCs, and that's why I covered this paper.
Yeah, let's just clarify that I don't think Paul's going to be happy about this paper because he's trying to make dog meat or anything.
No,
All right,
no, 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 no.
let's just be very clear about that. And yeah, I was thinking the same thing. Like, why are we doing this? And then, which not it doesn't matter what I think, first of all, but also it doesn't matter if there's a clear why. But nevertheless, I'm like, why? And then I was thinking like, oh, cloning, right? Because that was a thing with the Hwang Suk fiasco, and this is South Korea, right? So I'm not saying it's like a cultural thing at all, but there was definitely a silver lining there, which is that he landed on his feet cloning dogs for Barbara Streisand and all that. So I don't know, maybe the material need isn't there because of the SCNT will do it, but who knows? There's that in terms of generating stem cells that you could probably more robustly generate like genetically identical dogs. But looking into the introduction here, I found a better answer, which is that dogs, and it makes sense, dogs through the history of their breeding, and you see this, it's devastating. But they have all these really like enriched bad diseases. My in-laws have had a couple of dogs that like that more and more this happens. Dies dying these dogs that are from the breeders dying younger with increasingly malignant, terrible diseases. For example, the prevalence of dilated cardiomyopathy in humans, this is straight from the intro, 0.004%. So what is that? Like 4 in 100,000 or something in human. In Doberman pinschers, it's 58%, more than half of them.
Jeez.
So,
Wow.
like, we have this playground of enriched disease states that are really similar to human, comparable to human in many cases. We've been unintentionally creating this lab of model animals that approximate devastating and rare human diseases. So there it is that now we have a cell-based model where, in pursuit of that NIH new model of animal minusing animals from the modeling platform, this could be it. So, really strong rationale for making canine iPSCs. I'm glad I went in there, and clearly, again, not the first person to think of making these cells. So clearly, there's a bit of a lift here in optimizing conditions by whatever alchemy and observational turnaround. So kudos to this group and to you, Paul Burridge, who's clearly dancing in the streets with all the iPS cells that are being derived.
Yes, and to be clear, I'm Paul, I'm not saying you're gonna make dog meat from these iPSCs, I'm just saying as an
If you
aficionado,
wanted to, Yobi, I'm
I
still waiting for you to make some human meat. Who says there's anything wrong with that? I
Yeah,
mean, what is the problem with a little bit of benign cannibalism, Arun?
I can name a few things, but yeah, Paul, you may wanna drop off the show at this point. You've
Yeah.
got enough to worry about right now.
Yes.
But no, I'm glad you clarified the application for dog disease modeling. Yeah, no, for sure. I mean, that number is insane. 53
Crazy.
58%
And that's
of
just
that
one.
speed.
That's just one breed. I mean, there's a lot. There's a lot, and a lot of them get sick. It's so sad.
Yeah,
Um, yeah.
yeah, and just real quick, I mean, not just the cardiomyopathy, but also all the musculoskeletal problems, especially that the big dogs have. So maybe you can model some of those using these iPSCs too.
Them big dogs. Woof woof. Anyway, it's a good day. I wouldn't say it's a good day for the dogs. I still feel for them. They're not getting a lot out of this, but uh, there's some upside for sure for human health, and a lot of creative applications here today. I loved the way we handled our stories today, highlighting what an effort it is. And honestly, these stories should inspire you. It's hard for all of us. So whatever problem you're banging your head against, keep at it. Think different. Try something else. We'll talk about more of that and some just basic curiosity based research and driven hypotheses with Carole LaBonne in just a second. Before we get there, quick message from Stem Cell Technologies, who invites you to find a pathway into the peripheral nervous system with Stem Diff Neural Crest Differentiation Kit. Whether you need Schwann cells, sensory neurons, or sympathetic neurons, Stem Diff Neural Crest Differentiation Kit enables high purity generation of neural crest precursors from human pluripotent stem cells. Learn more at www.com/NCKIT. All right, everybody. With us today, we have Dr. Carole LaBonne, who is past president of the Society for Developmental Biology and the Erastus Otis Haven Professor of Molecular Biosciences at Northwestern University. The LaBonne Laboratory has been understanding the genesis of neural crest stem cells at the level of the signaling pathways and transcription factors that comprise the neural crest gene regulatory network. They also study how these mechanisms contribute to exit from pluripotency and the subsequent lineage restriction of neural crest stem cells to their derivative cell types that collectively define vertebrates. This is big science, guys. Carol, thank you so much for joining us today on the show.
Happy to be here.
Yeah, thanks so much for being here. So, I guess Dalen kind of summarized it. Your lab studies the beginnings of neural crest stem cells, their retention of pluripotency, regulation of pluripotency, all that good stuff that we're familiar with here on the show as pluripotent stem cell biologists. So, I guess could you give us an overview in your own words of what your group is focusing on right now and perhaps what you're most excited about when it comes to neural crest biology?
Sure. I mean, they are without doubt the coolest cells in developmental biology. They are the cells that make you not a sea squirt. So our closest relatives, non-vertebrate chordates, our sister outgroup are the tunicates. Siona incestinalis is the one that people study most in the laboratory. Uh, we would still be those kinds of sessile, filter feeding invertebrate, what we call wet squishies, if it weren't for 500 million years ago at the base of the vertebrates having acquired this new stem cell population. So neural crest cells contribute so many important cell types to the body plan. They give us our facial structures that allow for predation rather than filter feeding. They've allowed us to evolve a larger brain because we have a skull to protect it. They allowed us to be bigger, more successful critters because we have a peripheral nervous system to take in sensory input from the periphery and relay it to that new brain. They give us pigmentation for sexual dimorphism and camouflage, and a sympathoadrenal gland that allows a flight or fight instinct. So basically, everything that collectively makes you not a sea squirt are derived from these cells. And so, if you want to understand where we as humans or other vertebrates come from, understanding how these cells formed and develop is absolutely essential. They also underlie a large percentage of birth defects in live births.
Yeah, I mean, you're taking me back because I recall first learning about neural crest stem cells and thinking how they're just like this jack of all trades and have this incredibly broad developmental purview. They're so difficult to characterize and silo. And we're going to come back to that in a minute. But you're taking me back because when I learned this was when I was literally in my first week of grad school, when I was introduced to Xenopus and spent the next couple of years squeezing eggs out of females and watching these fertilized embryos progress to swimming tadpoles in like 36 hours right before my eyes. I mean, it was a real great immersive experience to draw in any individual, much less someone who has a inkling of an interest in science. I'm totally captivating. But that, accessibility and transparency of the Xenopus embryos and other amphibian models enabled some of the first lineage tracing studies that elucidated and explored the neural crest way back in the mid 20th century, right? So yeah, I mean, and hearing you talk about it, I could see what we're talking about is big science, but big basic, right? And nowadays, there's so much focus on clinical translation. We sometimes fall into this trap of defining the value of our experimental values as a function to how closely they approximate human biology or physiology. But I personally, I miss the microinjection, animal cap assays, it was so satisfying, gratifying, and allowed you to ask really unique questions with an immediacy that I think still to this day, these more sophisticated or closer to human models don't really get at. Although maybe notwithstanding organoids, which really changed the game. But for all the listeners out there who woke up in science with human embryonic stem cells already having been derived and may look askance at any animal model outside of non-human primates, maybe make the case for Xenopus as someone who uses it in the modern landscape.
Sure. I mean, they kind of make the case for themselves, to be honest. So you already mentioned that they develop in simple salt solution. So that allows you to have access to them throughout their entire development. And sure, that's true of organoids as well. But we still don't know that organoids are accurately reproducing what happens in vivo. And the only way that we know that is by studying animals in vivo and then asking, are organoids replicating what we know to be true? So Xenopus, the kinds of strengths that it has, my personal favorite is the first cleavage divides them into left-right halves. So you can do an experiment on one side of the embryo and you always know what normal development looks like because it's the other side of the embryo. And so that allows you to get at very subtle phenotypes that you never would be able to see if you were comparing pups from the same litter or even pups from a different litter, if you're talking about mice, etc. So that's an enormous advantage to the system, as is how fast they develop, right? So, and they were actually the first embryonic stem cells, those animal caps that you mentioned in passing, right? So all jawed vertebrates, and now recent work from my lab, we've shown all vertebrates have pluripotent stem cells at the blastula stage. And in Xenopus, you can take those out very easily. Since you're culturing them in a simple salt solution, you're completely in charge of what they see and when they see it. And so you can direct them to form any cell type in the embryo just as you can with a mouse or a human ES cell. But they are the real deal, right? So pluripotency endogenously is a transient state.
Mm.
In mouse and human ES cells, we're retaining that artificially. And so again, you're not always certain that you're actually replicating what happens in vivo. The cool advantage of the Xenopus system, too, is how fast they move from the top of Waddington's landscape to the bottom. So from pluripotency to lineage restriction. Mouse or human ES cells in culture, that's way more than a week. 7 hours for Xenopus. And so that allows us to go and follow the gene regulatory changes, the epigenetic changes as cells transit from pluripotency to a lineage restricted state with incredible focus on the dynamics of the process that you just absolutely couldn't do in a mammalian system.
Yeah, it's an incredibly powerful system, and we've covered it, I wouldn't say extensively, but we refer to it often here on the show, in addition to some of my other favorite vertebrate model systems, including the zebrafish, for example, which of course have a similar power in that, they're scalable, genetically very malleable. They have an ease of imaging as well. It's a good point, and I think perhaps a point that's lost on a lot of junior trainees that human embryonic stem cells, iPSCs, of course, are entirely artificial.
Yeah.
These cells don't truly exist in vivo, whereas in contrast, in Xenopus, there are true pluripotent stem cells that you can manipulate, you can identify. So, yeah, there's pros and cons of in vitro versus in vivo systems, and we talked about that a lot here on the show. But there's a lot we can learn from this really cool model system. And one thing we've talked about recently a lot here on the show is direct reprogramming. So the ability to go from one cell type to another. For example, I'm a cardiac biologist, and there's been studies where you can go from fibroblasts directly to cardiomyocytes. It's not a very efficient process, but it's been explored. And also recently, there's been a growth in the use of small molecules to actually drive this process forward. And you've actually done some similar work where you showed that small molecules can reprogram Xenopus blastula stem cells into neural crest fate. You had a recent paper on that. Bypassing genetic manipulation is, quote, a cleaner process. And like I said, I think this is pretty relevant given what we talk about here on the Stem Cell Podcast. So tell us more about that work, how you came to test that chemical approach, and what insights might it reveal about the mechanisms of neural crest induction.
Sure. So again, we were really became interested in those pluripotent blastula cells because we had found that the gene regulatory control of pluripotency in those and in neural crest stem cells have remarkable overlap. And so we developed this platform. I mentioned that 7 hour transit from pluripotency to lineage restriction, where we could really follow the changes dynamically. And what we really wanted to do was to follow the changes from a pluripotent blastula cell to a neural crest stem cell But the ways that we had for generating neural crest were less satisfying than the ones that we could use for some other cell types, because it either meant we had to inject messenger RNA, let's say for Wnt to upregulate beta-catenin signaling, and cordon or noggin to downregulate BMP signaling at the two-cell stage, which means you're not starting the experiment at the pluripotent blastula stage, you're starting it earlier. Or we could use hormone-inducible constructs for transcription factors, but that meant that you were going to have heterogeneity in the cells that were expressing them. So what we wanted to do was to start at the blastula stage and be able to use small molecules to induce neural crest cells. And so we just, basically, the work of a postdoc, Paul Huber, we tried out different Wnt agonists and BMP inhibitors and different doses, and came up with a really robust system where we can generate a pure population of neural crest cells. And Paul actually put these through their paces by taking grafts of these and putting them back into the embryo and showing that they could give rise to all of the neural crest derivatives. In fact, one really beautiful experiment that he did was to show that you could have the entire craniofacial skeleton on one side of the embryo, because it did a unilateral grafting, entirely made up of the in vitro induced stem cells.
Yeah, getting into neurocrest, and you alluded earlier there, and I think a connection you just drew as well to the craniofacial abnormalities in terms of like the clinical endpoints, right? But like taking it back to the basic biology, is the cell biology of it, and kind of looping in what Arun was mentioning about direct reprogramming, you know, iPSCs and then direct reprogramming kind of was, broke the dogma that there was this unidirectional differentiation, right? But neural crest, I think, was maybe kind of OG emblematic of the plasticity that's manifest in normal development, right? Although in my early training, the one of the central dogmas is that the three germ layers were very neatly siloed. But like in the intervening time, I'm not going to age myself here, but it's
Ha
been
ha ha.
a couple of decades. Um,
Better to age you than me.
well, let's say it was about only 10 years ago, roughly, although the idea has kind of circulated, but I think it was around 10 years ago in this Dev Cell paper, that the authors are escaping me right now. There was like definitively a revision of that thinking to incorporate this observation of like neuromesodermal progenitors. So, like, there has been this whole conversation just about the basic cell biology and epigenetics and kind of, the whole Waddington idea and crossing over between lineages that makes these cells super fascinating from a cell biological perspective. But also, loop going back to the clinical endpoints. We recently talked on the show, and recently being like a couple years ago when the paper came out, because at the stem cell show, we were talking about these human pluripotent stem cell model by Lawrence Studer's lab, where they generated vagal and sacral neural crest cells that really, vividly and impressively rescued this model of Hirschsprung disease. So the idea of neural crest cells having clinical applications, it's real and emerging. Are there other aspects, other clinically focused endpoints in your lab, that you could elaborate on?
Well, yeah, I mean, as you noted, I mean, it's actually greater than you think. There are estimates that even up to 90% of birth defects in live births are neural crestopathies. And that kind of makes sense, because if you think about what I was saying about building a chordate body plan and then layering on all the bells and whistles that make us vertebrates, when you have defects in that sort of core chordate body plan, you're almost never going to have a live birth. Whereas a lot of the things that neural crest cells give rise to, the craniofacial skeleton, the pigmentation, uh, parts of the enteric nervous system, etc., these are the ones where you can have a live birth, right? And so that gives us the ability going forward to be able not only to identify the genetic basis for human birth defects and syndromes, but also now with the advent of CRISPR therapies, the potential that we might actually be able to correct some of these. And so I think that's incredibly exciting. But beyond what you were just talking about with respect to birth defects, there's also a lot of cancers that are neural crestopathies that are neural
Mm.
crest derived. So neuroblastoma, which is the most solid pediatric cancer, melanoma, which is a cancer of your melanocytes, right? And so by understanding neural crest cell biology writ large, we also gain a lot insight into a lot of cancers that are critically important to be able to understand.
Yeah, I mean, the extent of it, just it's not just, every cell. It seems like that these neural crest stem cells make it across the body plan, but yeah, they're implicated, perhaps, not surprisingly, in a lot of disease mechanisms. So I wanna shift gears a bit here, because you've been a outspoken advocate. I mean, we're going a bit to a third rail here, but it needs to be said, and you're in multiple leadership positions that I think empower you to speak out with courage about these things. And you've been an outspoken advocate along those lines for many things, but including curiosity driven basic science and its role in both maintaining scientific leadership as well as seeding future innovation in the US. And in recent weeks, months, years, political battles are having a pretty outsized impact on the practice of research and inquiry. I mean, on the practice of a lot of things, medicine, but more, it's, focus on what we talk about on the show. It's really impacted research. And it remains to be seen how extensive the casualties will be when the dust settles. Um, but perhaps more concerning to me are the long-term consequences, which really is an unknown there. But you're someone who's right in the middle of it. I mean, your funding has been jeopardized. We spoke before the show how you have a daughter who's a first year PhD. I mean, you're on so many sides of this, so you're not afraid to talk about it, and I think take the lead. So give us your take on the impact that you think is forthcoming, long term, short term, and what you think can be done to get things back on track.
So I think even before the this year and the dramatic politicization of science, we saw inklings of this idea that we're done with basic science and all we need to do is translate. And that's so misguided because there's so much that we don't understand, about developmental biology, which is what I'm passionate about, stem cell biology, but also about all other areas of biology. We are nowhere near done with studying basic science. And it's really important, and what I think the general lay public doesn't understand, but even some scientists don't engage with, is the fact that most of the advances that happen in the clinic did not come from studies that were trying to cure a disease or intervene in a particular syndrome or what have you. They came from decades earlier basic science. And that's true of drugs as well, right? So this is far out from my area of study, but everybody is big into Ozempic and these GLP-1 inhibitors, right? But they derived from studies more than a decade ago, studying the Gila monster, which is a lizard that lives in the desert that has this amazing ability to fast. And so somebody was just curious about how they could go so long without food. And they isolated a compound from the saliva that eventually led to all of these drugs that are now changing people's lives, right? But had someone not had curiosity about a Gila monster, we wouldn't have all these drugs that are helping with diabetes and obesity and many other things, it turns out. So basic science is so important. It is what drives the later translational and the later applied discoveries. Without it, all of those will eventually dry up, right? So it's critically important because the investments that we make today are going to be the cures of 10, 15, even 20 years from now. And so we basically cut off that pipeline if we stop investing in basic science. And in terms of pipeline, I mean, the pharmaceutical industry and the biotech industry, they translate the basic discoveries that are made in university labs and that are funded by things like the NIH. So they can't do their job because they don't do the basic science. They rely on us to do the basic science, and they rely on us to train their workforce. So their workforce of PhDs will dry up without the investments in university-based science, including basic science. So this is of critical importance, and a huge percentage of the US economy is driven by investments in non-military research. In science research and biomedical research. And so the economy will take a huge hit. It's been estimated to be as big as the hit that we took in the Great Recession if we stop funding science at the university level.
Hmm.
You're right, I'm passionate about that.
No, very, very well said. Yeah, basic science is important. It matters. And I think part of it is a failure of communication, not just within the scientific community, but outside as well to convey the importance of basic science. Because, yeah, the general public sees, oh, something like, I'll just give an example of CRISPR, right?
Yeah.
CRISPR genome editing, which has such an amazing translational impact over the last decade. That didn't come out of thin air. Came out of 30 years of work from Doudna, Charpentier, and colleagues, figuring out why these bacteria have these antiviral defense mechanisms that back then no one really quote, cared about, right? But now everybody cares about it because you can edit DNA really super quickly, cheaply, easily, whatever. So yeah, basic science absolutely matters, and we gotta continue supporting it.
Yeah,
And
the CRISPR is a great example because, again, they weren't even looking for how they protected themselves from viruses. Initially, it was just pure curiosity of why are there these repeats in the genome,
Hmm.
right? So, what pharmaceutical company is going to say, "Oh, cool, that bacteria has repeats in its DNA. Let's figure out why," right? But yet, you're right. I mean, we just saw a baby cured of a disease using CRISPR, and that's just the tip of the iceberg of what's to come.
Hmm.
Absolutely. And I mean, you're fortunate enough to be at a place like Northwestern that has amazing resources in basic science, right? But I mean, obviously, Northwestern Hospital is a great translational environment as well, great clinical applications there. I've got some mentors like Paul Burridge, who we've had out here on the show, who have had great success at Northwestern. So, tell us about what it's like to be there. Obviously, it's a crazy environment right now, but what have you enjoyed about working at there?
I was gonna say you gave me a good belly laugh there because no one these days is saying it's a good Northwestern is a great place to be. For your listeners to don't know, we have had all of our federal funding frozen since March, and so basically no NIH money has been flowing into Northwestern for a long time. Let's just say 6 months, and that has been enormously stressful. On the faculty, of course, but also on the trainees. And I'm really worried that we're going to lose an entire generation of scientists with all the hits that everything has taken and how scared these kids are about whether they're going to get jobs or whether they've made the right career choice. We're really going down a dangerous path right now. But Northwestern, before all of this,
Hmm.
a fantastic place to do science because, in part, because it sort of lives and breathes interdisciplinarity. So, I've had collaborators with mathematicians, with physicists, with engineers, with chemists. And at many universities, those folks don't even talk to one another. We share a graduate program and really a fervent belief that the best science plays out at the interface between disciplines and that by working together we can do things that neither one of us could do on our own. And so that makes it an exciting place to do science.
Yeah, and what you just drove home there, I mean, that's the big picture in those two answers, and with the belly laugh in between. But you have to laugh or you'll cry, right? Because we're talking about what you just described before, right? A place where you can have all these unique disciplines, and you can have young people who are enthusiastic, but maybe naive and not cynical, and they learn. That's what they learn at a place like this. They do the experiment, and they get that feeling of seeing something for the first time, and they learn the beauty of that. They learn how to ask questions and how to be curious, and that's what's at stake here in what you're describing. It's 6 months without funding. It's not just like that 6 months, but there's a lot of people looking over their shoulder like, "Oh, you know what? I was going to, but who knows? The future looks dark." Like, it's really, the impact of what feels like oftentimes arbitrary or misinformed decisions at the highest level, uh, can have these knock-on effects that lead to, as you said. in the worst case, a whole generation that just lacks the interest in curiosity-driven science, and we've lost that this whole latent potential that arises out of these unknown, I wonder why type experiments. So it's such a, I mean, you've said it so well, but I'm like, I got my hair raising the stakes here. I'm going to leave this conversation appreciating what we're able to retain here at our own institute, but also a little bit scared about what's coming. Um,
I'm gonna correct one thing that you said, if you don't mind,
oh,
which is
please, please.
I don't think this was accidental or unintentional.
Mm,
This what's going on right now with respect to science is very much intentional, and you can see it written out in Project 2025. It's a deliberate attempt to destroy universities at the highest level.
mm,
And the funding is one way that they can do that. The international students is another way. Financial aid is another way. But the idea is, and it's written out, that they want to eliminate places of independent thought and inquiry. And this is something that's happened in other countries like Poland and
mm.
Hungary.
Oh, well, this has really been stimulating in every way, good, but eye-opening in a shocking way. And I feel like you're saying the quiet part out loud and a call to arms, and I appreciate that. And I think our listeners will appreciate it as well. Before we let you go, though, we have a couple maybe on the lighter side. Uh, just a few questions, a couple to get to know you. First, what's the best piece of advice? I mean, it doesn't have to be light. Be real, girl. What's the best piece of advice that you've ever been given, professional or not?
Okay, so this piece of advice came primarily from my postdoctoral mentor, before she was my postdoctoral mentor, Dr. Marianne Bronner at Caltech. But when I first met her at the Marine Biological Laboratory when I was a graduate student, she was doing some sort of spontaneous mentoring of the young women who were there at the time for the course. And one of the things she said was, don't question whether or not you can have a career and children or when the right time is, just do it. So she's, her viewpoint was, there's no good time, there's no bad time, so you need to just let your choose when you want to plan your family and let everything else sort of fall out around it. And I don't know if she knew that I would take her quite so literally, because I had two kids during my postdoc with her. She likes to claim that I was pregnant for the entire time I was a postdoc, and I think it was really only about 55%. But point taken. But I try to express this to all trainees, but particularly the female trainees, that they're actually, if you want to have a career, not a job, there actually is no better career than academia to combine with having a family and being a parent or being a mother. And so, I think there's a lot of people who sort of say that's not the case, but they're just wrong. And I think that being a parent has made me a better lab head, and being a scientist has made me a better parent.
Yeah, I mean, it was easier. The path was easier for me, but parented has been the best part of my life. And talk about a productive postdoc. I mean, forget about the papers. You're
Or a
churning.
reproductive postdoc.
Reproductive. You made my joke even better. All right. Finally, what is the biggest misconception about science that you would like to resolve?
So I kind of hate on how we teach science in this country, particularly biology, but science writ large is sort of memorizing facts in a book, which makes it really just unappealing. We don't do a very good job of instilling in kids what science really is, which is a process of inquiry, right? I mean, as you mentioned earlier, you discover something new. It's that eureka moment. It's exciting. And so there's that, but also the idea that gets pushed sometimes, that if what we think we know about a process changes, that means there's something wrong with science.
Hmm.
And that's absolutely flipped on its head. It means there's something right about science, right? We don't discover something and then leave it there set in stone and go look at something else. We keep pushing at it. And sometimes we build depth in the things we've already discovered, and sometimes we nullify whatever our hypothesis was. But that is is the process of science, and that means that science is working, not that it's not working. And I think that sometimes some of the negative attention that it gets focused on science is because of these changes, as if somehow that means that we're sloppy or that there's wrongdoing, when it really just means that as new techniques and new information becomes available and we turn around and shine that spotlight on old problems, we're going to learn more.
Hmm. Yeah, speaking of the light, I'm thinking of the everyone says, "Oh, the Thomas Edison, he didn't fail 10,000 times. He found 10,000 ways not to invent a light bulb or whatever." But what's not said there, as you just alluded to, is that then people keep, better mousetrapping that, right? And
Yep,
that's how you get to whatever LEDs. I don't know where we are now, but lasers, all of it.
yep.
You keep improving upon, you keep questioning the advance to find the truth underneath the truth. So I'm glad you mentioned that. That is such a huge part, probably 90% of research in science. I mean, what an arbitrary number, but it's what it feels like to me. Anyway, Carole, this has been such a great conversation. Thanks for all the insights and for speaking out. I hope our listeners gained from it. I know that Arun and I did. We appreciate you coming on and sharing with us.
Thanks so much, and thank you guys for doing this. I think it's a great program and a great idea.
Well, thank you. That's very kind of you. All right, y'all. That brings us to the end. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at Stem Cell Podcast, Blue Sky at stemcellpodcast.com, or via email at info@stemcellpodcast.com with feedback or to suggest guests. That's another one in the books, you guys. Thank you so much for listening. We'll be back in your ears in a couple weeks.